Welcome, and thank you so much for joining us again today. We get to talk about homeostasis, and we're going to throw in some scientific method stuff too. So, super important day, and keep calm, maintain homeostasis, and remember that science rocks.
Once again, it is super important for you to create a biology notebook, whether it's online in Google Drive or a spiral notebook. Take notes. Copy down these 27 vocab words. Remember, they go in ABC order like this. And come back later and create your own definitions of these words. Use your own words. So go ahead and pause this if you need to finish writing down the terms. Okay, homeostasis. It's the regulation of metabolic processes to maintain the stable internal conditions required for survival for an organism. The body has mechanisms for maintaining a stable internal temperature, pH balance, and ion concentration. These mechanisms often require several organ systems to interact with one another in order to maintain homeostasis. Onto the cell membrane and homeostasis. One of the features that allows cells to perform their necessary activities is their selective permeability or semi-permeable cell membranes. These membranes are located beneath the cell walls of plant cells, and they form the outer boundary of the animal cells. So it would be like this dark blue layer here. The semi-permeability of the membrane permits cells to control what comes into the cell and what goes out of the cell. So if you were to take this outer cell membrane, and so this outer blue part, and blow it up, it would look like this. Okay, all these little parts. So this is the cell membrane or the plasma membrane blown up. A structure called the lipid bilayer makes up the vast majority of the cell membrane. So a lipid bilayer is talking about these phospholipids, how here's one layer and here's another layer. So bi means two, so it's got two layers of these phospholipids. The lipid bilayer is formed from the two layers of the phospholipids. So like here is just a simple model of the lipid bilayer of the phospholipids. So one phospholipid would be like, here's the head and then its little tails. So that would be one phospholipid. It has a unique structure. The hydrophilic, it means water-loving, hydrophilic, heads of the phospholipids. So here's the heads, the circle parts. In this model, they're orange. Okay, they face outward, so toward the exterior and interior of the cell. Because, okay, outside the cell is mainly water, and inside the cell is cytoplasm, which is a lot of water, and it wants to be attracted to water to easily flow around. So the hydrophilic heads are attracted to water. So they're going to be on the outsides. In the very inside of the plasma membrane, it's called hydrophobic. Okay, think fear, phobic, water fearing, hydrophobic. So these tails face inward. So notice these little transport proteins, these purple things here. That's going to allow substances to flow through the plasma membrane. The structure and function of a cell membrane is described by the fluid mosaic model. According to this model, the membrane is like a mosaic, think art, mosaic, because it is made up of many different parts. Okay, think of like a tile or like a stained glass made up of many different parts. That's the mosaic part, including different types of carbohydrates, okay, different types of proteins, different types of lipids, all right? And also, according to this model, the membrane is considered fluid, remember fluid mosaic model, because the structures within the membrane can move around. Remember we talked about the phospholipid heads that are attracted to water, so it can easily move around. Water is an essential component of plant and animal materials and crucial for the maintenance of homeostasis in the cell. It provides a soluble environment for chemical processes, serves as a reactant in some chemical reactions, and provides hydration that maintains the cell shape. Osmosis is a type of diffusion that specifically involves the movement of water only across a selectively permeable membrane. So, during osmosis, water moves from areas of high concentration to areas of low concentration. So, energy or ATP is not needed or used from the cell to do this. So, in this model, you can see that just the blue little water molecules, so here's the blue water molecules and the red are the sugar. So, just the blue little water molecules are moving across this selectively permeable membrane. Not the bigger red sugar molecules, just the water molecules. It's like a puzzle, and just the water molecules are able to fit through. So that's osmosis. Osmosis, water. Okay, we're going to talk about 3 different types of solution that the cell could be in: isotonic solution, hypotonic solution, and hypertonic solutions. We're going to start off with isotonic solution. Iso, the prefix means same. So the concentration of dissolved substances and water outside the cell is the same as the concentration of dissolved substances and water inside the cell. So if you look at this first model here, it's just like the previous key, where water molecules are the little blue dots and the dissolved molecule, it could be sugar, it could be salt, are the bigger red ones. So if you notice this cell, and H2O is water, so we've got water moving into and out of the cell at the same rate. Look at the thickness of the arrows, they're the same. Okay, so if you think of the same concentration, I always think of it as like Kool-Aid or lemonade. You're making some in a pitcher of water, and sometimes people get confused between concentration and amount. So if you were to make some lemonade and it was in a pitcher of water and you poured yourself a glass of lemonade, okay, the lemonade that you're drinking from the glass is going to taste the same as the lemonade if you were to pick up the huge jug and drink out of the pitcher. Okay, it would taste the same. They have the same concentration, same amount of water, same amount of sugar, same amount of lemon. It might be a different amount because your glass of lemonade is a smaller amount than the entire pitcher of lemonade, but they taste the same. Okay, this is kind of what this is talking about. Same concentration. And if you look at this animal cell and plant cell, notice the arrows again. Same amount of water is going into and out of the cell, a normal, constant rate. If you look at this picture of a plant cell, it has a normal shape and pressure. It's the rectangle shape. The cell membrane looks normal. The vacuole looks normal. Everything is happy. The next type of solution a cell could be in is hypotonic solution. So the concentration of water molecules is higher outside the cell than inside the cell. Okay, so inside the cell needs water. So look at this model right here. Look at the inside of the cell. See how many dissolved molecules, let's say they're sugar, the red ones, are inside of the cell. Okay, and look at the concentration of water outside of the cell. There's lots of blue dots. There's lots of water outside of the cell. So the concentration of water molecules is higher outside of the cell. If you always remember, water moves to the area that needs more water. Okay, so the inside of the cell is like totally salty or totally sugary, all this red dissolved molecules. The inside of the cell needs water. All right, look at your arrows. H2O is water. The arrow going inside of the cell is a lot thicker than the arrow leaving the cell. There's still going to be some water that leaves the cell. Notice the thickness of the arrows. But if a cell is in a hypotonic solution, most of your water is going to want to gush into the cell because the inside of the cell needs water. All right, so water molecules enter the cell by osmosis. Notice these pictures of the animal and plant cell and your big arrows going into the cell. Remember, some is still going to leave the cell. In this last picture of the plant cell, look at that. The cell swells as pressure increases. So all this water is going into the cell. The vacuole gets huge. The cell membrane is like pushing up against the cell wall. I remember it. Hypo kind of sounds like hippo, and hippos are huge, right? So if a cell is in a hypotonic solution, okay, and if water keeps moving into the cell, it's going to be like a hippo, humongous, and hopefully eventually it will level off and get back to that isotonic rate. Okay, the last solution a cell could be in is hypertonic solution. The concentration of water molecules this time is higher inside of the cell than outside of the cell. So the outside of the cell is the area that needs water. Okay, look at the cell. There's all these water molecules inside of the cell. All right. And outside of the cell, it's like totally sugary or totally salty. It's got all these red dissolved molecules. The outside of the cell needs water. So notice your arrows. Water is gushing out of the cell at a fast rate, and water is a little bit going into the cells still. Okay, but most of your water is going to leave the cell because the solution around the cell needs water. All right, and look at your animal and plant cell here. The arrows are leaving. The water is leaving the cell. Remember, a little bit still goes in. In this picture of the plant cell, holy moly, the cell is shrinking and losing pressure, okay? You'll notice, okay, here's the cell wall, and the cell membrane, this blue part here, used to be all around this rectangle, but it's still connected a little bit, and it's getting the water sucked out of it, and it's totally shrinking. And if you do this experiment under a microscope and use some water and salt water, you can actually see this happen. Like, you can see the cell membrane shrink away from the cell wall, and it's awesome. I try to remember hypertonic solution like a hyper person, okay? They're always moving around. They're really fast. They're super skinny. Okay, not really, but it helps me remember what this means. Hyper, think crazy moving, super skinny, and all of the water is getting sucked out of the cell, super skinny. In order to maintain homeostasis, positive and negative feedback mechanisms are both involved in organism responses to stimuli. So, a positive feedback mechanism is an interaction that increases or amplifies the response of a system in which it's incorporated. So, blood clotting is an example of positive feedback. So, you have this damage, this cut, this damage to a vessel wall. It'll signal for the release of substances that trigger the activation of blood platelets. So, yay, blood platelets. As platelets begin to accumulate around the damaged portion, they send chemical signals to activate more platelets, resulting in the formation of a clot, which acts as a temporary plug or like a scab that this person's picking at until the vessel tissue can be repaired. And then gross, you pick it off again. Oh, look, start over. So, positive feedback mechanisms. A negative feedback mechanism is an interaction that reduces or dampens the response of a system in which it's incorporated. So negative feedback loops are actually more common mechanisms in maintaining homeostasis, whereas positive feedback loops pretty much reinforce changes to a system. So negative feedback loops are responses in which a system works against a change. So the insulin production model best explains a negative feedback mechanism. So you have these beta cells in the pancreas which release insulin into the blood. Some of the insulin goes to the liver and some of the insulin goes to the blood. So your body cells become permeable to this glucose. Okay, and the blood glucose level decreases. Remember, negative feedback, so the blood glucose level decreases to this normal level. So you've got this homeostasis blood glucose level, and then you eat. Okay, so after a meal, blood sugar level actually rises, so it gets higher. Okay, and then your beta cells in the pancreas release more insulin into the blood. Okay, the liver converts it to glycogen, and some of the insulin goes straight to the blood where it becomes permeable to the glucose. Then it drops again. Blood glucose level decreases, and then you eat, it goes back up. Okay, so negative feedback mechanisms. All right, let's talk about the scientific method for a little bit. The scientific method and graphing will be dispersed throughout any topic in biology. So it's super duper important to know about both. The scientific method, let's just go through the steps. And I have 6 main steps for you right here. The first step of the scientific method, so say you're doing an experiment, you want to, number 1, make initial observations around you. Look around, okay? What's going on? What can you do? And then ask a question, okay? Whether you're in the field or whether you're in the lab, you want to look around and see what's going on. What can you even do an experiment over anyway? And ask a question about it. And then number 2, so after identifying a testable problem, then conduct research, okay? Get online, look some stuff up. And then number 3, create a hypothesis. And a hypothesis is often put in an if-then statement, okay? So if blank, comma, then blank, okay? So if I add fertilizer to a plant, comma, then its plant height is going to increase. All right, so if I do this, then this is going to happen, and it's what you think is going to happen in your experiment. And then number 4, conduct the experiment. All right, and then number 5, record the results and data. And often this is put in tables and graphs. And number 6, analyze your conclusions. What were your findings? All right, the scientific method, know it, love it, learn it. Dry mix. Dry mix. Dry mix is one of the most important things to learn, and it will get you through a lot of biology questions. So, dry mix. Dry mix means pretty much nothing on its own. So, what does it mean? Dry mix. Dry mix, the DRY in dry. The D stands for the dependent variable. The R is the responding variable. They're the same thing. So the dependent variable can also be known as the responding variable. That's how it responds. And you're always going to find the dependent variable on the y-axis. Okay, so the y-axis, and if you get confused which one is the x and which one is the y-axis, the y-axis is on this side, and I always remember it because it has this tall y-axis part, okay? And then your x-axis is going to be down here, all right? So in dry mix, it helps you remember which variable goes on which axis. So your dependent variable, how it responds, is always going to go on your y axis, dependent variable. All right, your independent variable is going to be the mix part, MIX. Your independent variable is also known as the manipulated variable. It's the one that I manipulated, like I did this, independent variable, and it's always going to go on the x-axis, okay? So the one that I manipulate in an experiment, I did this, independent variable, goes on the x-axis for your independent variable. So let's look at these. Let's look at this first one here. The effect of fertilizer on plant height. Okay, and this is kind of like our if-then statement for the hypothesis. If I add fertilizer to a plant, then the plant height is going to increase. So we're going to do this experiment over it. Remember dry mix, let's write this up here so we don't forget. Dry mix, okay. I added the different amounts of fertilizer in grams. Remember to always put your units in parentheses. So I added 0 grams of fertilizer to this plant. I added 5 grams. I added 10 grams. I added 15, and so on. I did this. I know this. It's what I manipulated. It's your independent variable on your x-axis. Okay, so what is the experiment measuring? How did it respond? What is its dependent variable on the y-axis? So you want to measure the plant height, and it looks like we're using centimeters. Okay, so after, let's say, 2 weeks, if that's your experiment, you're going to measure the effect of this fertilizer on plant height. Okay, because remember, we want to only change one variable, and our variable that we're going to change is the amount of fertilizer. We want to use the same plant, the same amount of soil, the same amount of sunlight. We want to measure it like the same day. Okay, so the variable that we're changing, that I manipulated, is the amount of fertilizer that we gave the plant. Okay, so when we did our measurements, it looked like and this is a bar graph, it looked like the plant that I gave 0 fertilizer to grew, all right, maybe 12, let's say, centimeters tall. Okay, the plant that I gave 5 grams of fertilizers to grew to maybe 14 centimeters tall. Okay, so the perfect amount of fertilizer, if you're wanting your plant to grow super tall, looks like would be 25 grams of fertilizer, because that plant grew about 21 centimeters tall. Okay, so there's all sorts of questions you could ask about this, but as long as you know how to correctly set up the graph, you'll be ahead of the game. And make sure you set up your graph in even increments. See how here they go by 5s? Okay, on the Y, they also went by 5s. They could have gone by 3s or whatever, but as long as it's by even steps, even increments, that'll be perfect. Okay, let's look at another example. How about change in Jim's heart rate during exercise? Okay, so remember dry mix, we'll write it down here. Okay, and when you do take a test, you could make notes and have a piece of paper and write down dry mix if you can, just to get that down on paper and in your head. So looks like the x-axis, what I manipulated was the time. So change in Jim's heart rate during exercise. I know that every 10 minutes I'm going to check Jim's heart rate. I am doing this. I know this. It's going to go on the x-axis. Okay, how did it respond? What is the dependent variable? That's going to be Jim's heart rate in beats per minute. Okay, and notice how they set up the graph, even increments. Down here on the X, they went by 10s, and then they went by 20s over here on the Y, and that's fine. So at 10 minutes, we checked Jim's heart rate, and that's when he started exercising, and it was 60 beats per minute. Okay, at 20 minutes, and this is a line graph, so at 20 minutes, they checked Jim's heart rate, and it was at 140 beats per minute. Okay. Same at 30, and that's when he stopped exercise. When he stopped exercising, his heart rate went back down. Okay, so after 40 minutes, his heart rate had dropped to about 75 beats per minute. Okay, and at about 50 minutes, it almost went back to its same start heart rate, okay, before exercise. So line graph, you would do your dots and then connect the lines, and just remember dry mix, and you can get it set up correctly. Okay, and there's one more example, more about height of plants, and this time instead of the bar graph, they used a line graph. Okay, for the heights of plants, and right here we're looking at. I know that every week, okay, I'm going to measure the plant height. Okay, so right here, the thing that I know is like the time. Okay, so I know on the x-axis that every week I'm going to measure how tall my plant gets, and this time we're using inches. Okay, so at week 1 is about a ½ inch, week 2 1 inch, week 3 3 inches, and week 4 your plant height was 5 inches, and there's your line graph. Okay, so dry mix, you guys, independent and dependent variables, super important. Make sure you know this. Here are a lot of other videos explaining more about these topics, some about the membranes and a lot about scientific method and graphing. So come back and watch them later and take more notes, or pause now and take a look at some. All right, let's do some practice questions. Number 1, how do the urinary, so we've got bladder, kidneys, so how do the urinary and circulatory systems, circulatory we have like the heart and vessels, so how do the urinary and circulatory systems interact to maintain homeostasis in humans? So let's look at these. A is saying the kidneys produce wastes that are carried within the bloodstream to the arteries where they are recycled into useful products. Okay, it's got kidneys in there, it's got arteries in there, it's got like the right parts, but the kidneys more like filter the blood. They don't produce waste. So I don't really like A. Let's look at B. The kidneys, so we're still in the urinary, so that's good. The kidneys transform food molecules into ATP. Okay, that's throwing me off right there. Not really talking about urinary or circulatory. This form of energy is carried by the bloodstream to muscles. Once again, not really talking about urinary or circulatory when activity is needed. So this one does not make sense. Okay, it's not, kidneys aren't transforming food molecules into ATP and talking about muscles. Doesn't really go with the question. So let's look at C. The kidneys produce growth hormones. Okay, growth hormones aren't really talking about urinary or circulatory systems that are carried through the bloodstream to parts of the body where they are needed. So I don't even like C. So I hope D makes sense. D. Blood flows to the kidneys. Yes, blood is circulatory. It does indeed flow to the kidneys, which is urinary. Cellular wastes are filtered from the blood, yes, and stored in the bladder, yes, which is urinary, until they are excreted from the body, yes, which is still talking about urinary. So thank you. D does indeed make sense. Number 2, the diagram below shows plant cells submerged in solutions with different concentrations of sugar. So we've got 1, 2, and 3 cells submerged in different concentrations of sugar. In diagram 3, so we're looking at this one, water is flowing by osmosis into the cell. Notice the arrows of H2O water flowing into the cell because the concentration of water is what? So the concentration of water is higher or lower. What are we talking about here? So remember we talked about hypotonic, hypertonic, and isotonic solutions. And remember that water always moves to the spot that needs more water. Okay, so in diagram 3, it looks like the inside of the cell needed more water. Okay, so the inside of the cell did not have a whole lot of water. The outside of the cell did have a whole lot of water. So the inside of the cell needed it, so it's going to go into the cell. So if we look at our choices, A is saying inside the cell is lower than the surrounding solution. So the concentration of water inside of the cell is lower than the surrounding solution. The inside of the cell needed water. So A actually makes sense. Number 3, in which of the following scenarios would a person experience an increase in blood flow? So we need an increase in blood flow. So let's look at these. A says surface blood vessels constrict in response to cold temperatures. Okay, constrict is like make smaller, and cold, burr. So it's making the blood vessels smaller. So that's not going to increase the blood flow. That's going to decrease the blood flow. So it's not A. B is talking about fats and cholesterol accumulate in the blood, which increases its viscosity. But when fats and cholesterol accumulate in the blood, it's going to block the passage, okay? So that's going to decrease the blood flow, not increase the blood flow. So that's not it. C, a person at rest begins to move heavy boxes up a flight of stairs. Okay, so you're just sitting there, and then all of a sudden you got to move all these boxes up a flight of stairs. So during exercise, cells require more oxygen and glucose, and respiratory rates increase in order to maximize oxygen intake, and the heart rate also increases in order to increase blood flow to tissues. So it's like you're starting to work out, and yes, it is going to increase your blood flow. So we've got C so far. Just to look at D, a factory worker cuts his hand on a machine and loses a pint of blood. Ouch. So that does nothing with increasing blood flow within your body. So C is the magical answer. Number 4, homeostasis refers to an organism's ability to maintain a relatively constant internal environment. So, sweating is a homeostatic process that humans undergo when it is hot. How does sweating help humans maintain homeostasis? So, how does sweating help humans maintain homeostasis? So, in order for a substance to evaporate, it must absorb heat energy, right? So if this substance is water, like sweat, it must absorb a lot of heat energy. So since water has a very high specific heat, body heat is absorbed by sweat and then transferred to the air, which cools the person off. So this is actually going to be B. So you need to know a little bit about that. Body heat is absorbed by sweat and then transferred to the air, which cools the person off. So this process contributes to the homeostasis of humans by allowing them to regulate their body temperatures. Number 5, in the human body, when the blood sugar level rises, a hormone known as insulin is released by the pancreas. This hormone speeds up the movement of sugar out of the blood, so the blood sugar level decreases and returns to equilibrium. So this is an example of what? So if you remember when we talked about the feedback, positive and negative feedback loops, this was one of our examples. So this is an example of it's not a learned behavior or a natural selection or a gene expression. So even if you look at those 3, none of them really even make sense. So a feedback mechanism. So the regulation of blood sugar levels by insulin is an example of a negative feedback mechanism, if you remember, that has evolved in order to maintain homeostasis. Last question, number 6. When dry environmental conditions exist, guard cells close the openings in leaves to reduce the loss of water from the plant. So that's cool. This process is an example of a feedback mechanism that plants use in order to what? To absorb more solar energy, to maintain homeostasis, to produce more chlorophyll, or to expend their resources. So if you were paying attention at all, you would know that we've been talking about homeostasis and that feedback mechanisms go with homeostasis. You could probably guess your way through this one. Remember, homeostasis refers to an organism's ability to maintain a relatively constant internal temperature, internal environment. In many organisms, feedback mechanisms have evolved to maintain homeostasis. So there we go. So, after fully covering the topic of homeostasis, you should be able to describe ways to design or improve an investigation about the importance of homeostasis. Homeostasis is a process that regulates or balances an organism's cellular functions as external factors change, allowing the organism to remain alive. So, I challenge you to create a project, a presentation, an experiment, a model, and show your teacher that you can explain this.